champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to talk about the next topic that comes under principles of inheritance and variation, that is incomplete dominance, codominance and pleiotropy. Ma'am, pleiotropy is not given in the NCERT, but the concept is there and the questions have come, so we should not be missing it as well. Yeah, so this is video number 28. And children, what all did we cover in the previous session? We spoke about the terms, very important terms in genetics, about which you should be definitely knowing. Then we spoke about inheritance of one gene, as in one gene that is coding for just one character, probably height. That's all. No other characters, just one character. We spoke about tallness and dwarfness, alleles, dominant, recessive, lot of things, and also the two laws of genetics, law of dominance and law of segregation. And this particular lecture will be in continuation with that, which is why I put it in this order. Now over here, the number of questions that was asked the previous year was 6, and 6 into 4, my dear children, 24 marks. Can we lose it? No, we cannot. And also, if you want the PDF of all the notes, you can join our WhatsApp channel. Okay. Now, a question that I gave my little NEET aspirants. Tell me the answer for this. A few of you might have already commented, but yes, others. Let's see who all got it correct, and let's see who all has still trying. In hybridization, capital T small t, that is a heterozygous tall condition, and small t gives rise to progeny of ratio what? Chalo, let's fuse. So these are the parents. These are the parents. Now let's see the gametes. So it can be capital T or it can be small t, similarly small t, or it can be another small t. Now, if this and this fuse get capital T small t, if this and this fuse you still get capital T small t, if this and this fuse you get small t, this and this fuse you get small t. Agree? Now, two of them are capital T small t, two of them are small t. Two of them are tall because capital T is dominant over the small t, and two of them are dwarf because it is a homozygous pair. When two alleles, recessive genes are coming together, you call this condition, basically they show the recessive trait, that is here dwarfness. So it is 2:2. If you talk about the phenotypic ratio, tall and dwarf, then also it is 2:2. If you talk about genotypic ratio, as in how the genes are looking, so over here it is again 2:2. Yeah, 2:2, can I cancel it and write 1:1? Will it be wrong? No. And do we have an option like that? Yes. So let's put down the option over here. This came in 1999. Okay. Now, my dear aspirants, I think that you got it correct. Going ahead with today's topic, why doesn't it work whenever I want? Chalo, I'll do. So, a little throwback to what we were learning, that is law of dominance, because I thought that I should have given more clarity. Now, law of dominance says that characters are controlled by discrete unit known as factors. For example, height is controlled by a gene which would be coding for a protein, probably what? Growth hormone. Fine. But height is controlled by a gene. So, characters are controlled by discrete units known as factors. So, he didn't know it was known as genes back then. So, he used the term factors because it was put down by Gregor Mendel. Now, factors occurs in pairs. Yes, the genes basically occurs in pairs. There are alleles. There are alleles, children. So, for height, you have capital T and then small t. Capital T will be coding for enough amount of proteins that will be increasing the height of the person, whereas small t might not be producing enough amount of proteins such that the height of the person is not too much. It's just there. That person has not grown big. Okay. So, factors, they occur in pairs, and these pairs are known as alleles, children. Now, in a dissimilar pair of factors, for example, when the factors over here, the alleles over here, they are not similar, they are dissimilar, as in like this. Okay. Three conditions can be possible. Either it can be like this, where two dominants one are same. Either it can be like this, or then it can be like this. Now we are talking about this particular thing. In a dissimilar pair of factors, okay, one pair or one member of the pair dominates the other, that is the recessive. So this allele that you see here is dominant, And whatever action it is doing, for example, it is producing growth hormone, this is why the animal or basically the organism or basically the plant is growing in size. Whereas this one affects this one's protein, or basically this one's effect is overshadowed by the dominant one. So this one will be known as recessive. Either it is not producing any protein or it is producing very less protein. But since you already have an allele that is producing enough amount of protein, all these negative effects are overshadowed. Okay. Now, law of dominance is used to explain the expression of only one parental character in a monohybrid cross. What do you mean by monohybrid cross? Basically, I am taking one character tall, one character that is height. Okay, and in Mendel's experiment, what he did? He took homozygous tall and homozygous dwarf, and then he made to fuse them. Okay, when he fused them like this, it came to his knowledge that basically all of them were capital T's, small t. Okay. So, law of dominance is used to explain the expression of only one parental character. What is that one parental character that is shown over here? All of them children are tall. So, tallness is one character that is being shown in the first generation, and this is because of law of dominance, because each allelic pair, if you see, each of them have one dominant allele. This is the reason that the first F1 generation, all of them are tall. Okay. And this is known as a monohybrid cross because you're just talking about one particular character, that is height, nothing else. And expression of both in F2. Now, expression of both as in both the characters are expressed in F2. Let's see what happens in F2 then. So, in F2, I take one parent like this, capital T, small t, capital T, small t. The gametes are capital T or small t. Capital T, this is capital T, okay, and small t. When I fuse this, I got capital T. When I fuse this and this, I got capital T, small t. When I fuse this and this, I get capital T, small t. When I fuse this and this, I get small t, small t. Now, over here I see one is dwarf, children. Over here, one is what? Dwarf, because there is no dominant allele to overshadow its effect. Right? There are no proteins being produced, or there is no enough proteins being produced, which is why this particular plant is dwarf. If a dominant allele was there, probably this particular effect would not have shown, and hence you are able to conclude the statement that in F2 generation, this is first generation, but in F2 generation you see both the parents because of law of dominance. Here law of dominance is working because no one is there to dominate it, which is why it is dwarf. But over here you see there is an allele to dominate, which is why all the three are what, children? Tall, and one is dwarf. If law of dominance was not working, then probably this would not have been a case. Yes, and there is no blending as well. There is no blending. So, the plants that are getting capital T, small t, they are not medium heighted plants. They are still tall. They are still tall. So, according to Mendel, if the condition was this and if the condition was this, it's producing the same amount of growth hormones, which is why it is similarly tall. It is similarly tall. He said blending is not there. So that was the second law that he put, that is law of segregation, according to which basically the alleles, when they come together, they are not blending together. They are just showing their character. So this one will be showing its character, this one will be showing its character. Which one is dominant will overshadow the other one. Okay. And according to him, both of them, even it was homozygous or heterozygous tall, both of them were producing the same amount of growth hormone, which is why they are similarly tall as well. But what? With time, things changed. There were certain exceptions that came through the way. Okay, I have written the tall, whatever I explained, I just made it before. So, with time, there are certain things that came in light, and there are terms also which came in light. And those terms are like this: incomplete dominance, as in something is not getting completely dominant. Let's see what's that something. So, basically, in flowers of snapdragon, okay, in flowers of snapdragon, they had red flowers with RR. Okay, RR always resemble what? Red flowers. Fine. small rr, I am writing, they resembled white flowers. Okay. So what did the other people do? They crossed capital RR and small rr. So over here, what are the gametes? I get R, I again get R, still R and R. Okay. So basically, this is on one chromosome, this is on another chromosome of an organism. Similarly, on one chromosome, on another chromosome. Either of the chromosome can come. Okay. Either of the chromosome basically got passed to the gamete. Whatever chromosome was there, this was 1, this is 2, this again 1, this is 2. Either of them got passed in the gamete, and now gametes are going to fuse. So when capital R fuses with small r, you get capital R small r. Similarly, same thing happens, right? Capital R small r, capital R small r. So according to Mendel, if we go by Mendel, basically this R is a dominant trait. Okay, so this should overshadow the effect of this, this should overshadow the effect of this. Again, the same thing should happen. But instead of all of them being red, all of them turned out to be pink. Shocked? Even Mendel could have been shocked if he got to know this. So basically, all of them were pink. Now people were like more curious. How can that be possible? We have to self-pollinate. So they self-pollinated this, that is, the progenies among themselves, they self-pollinated. So now we have the parents like this. So this is F1 generation, my dear children. This is F1 generation where all of them were pink. Now, okay, so over here the gametes are capital R, or it can contain small r. Similarly over here it can contain capital R or small r. Now let's fuse. This and this fuses to form capital RR. This and this fuses to form capital R small r. This and this fuses to form capital R small r. This and this fuses to form small r. Okay. Now let's see what was the conclusion. So capital R we know this has to be red flower. So again this became red. Okay. But these two guys, they are heterozygous, they are not homozygous, they are heterozygous, and now they are again pink. And over here small r, as we know, they are white. So in F2 generation, they were shocked to find this thing, that again red has come, pink is there and white is also there. We started with red and white, we didn't get red and white over here, instead we got pink. Now when we crossed, we got to know that there is red, pink and white still left. Law of segregation basically that has happened probably. Okay. Now but the thing over here that is very curious to understand is that why is pink color being formed over here? Why is it not red? Isn't R completely dominant on the other small r? That was the question. And the answer was yes, it was not completely dominant. So probably when this condition was there, okay, when double R condition was there, it was having enough amount of protein to produce what? The red color, children. To produce what? The red color. But in this condition where there is only single R, no enough protein to make it red. Instead, it was an intermediate thing that they saw. There was no enough protein to make it completely red, which is why I say it is incompletely dominant. It tried, but there is no enough protein to make it red. Okay, complete red. So basically it tried but didn't get success. Similarly over here, as you know, there is no one to try for red coloration. So it turns out to be white itself. So that is not a problem. But over here, as you see, it's showing incomplete dominance. Whereas when we spoke about pea plants, there, both of them were producing growth hormone and the plant was growing properly. But here in the case of snapdragon, things were quite different. And this term or this exception that has taken away from the law of dominance, usually this has to be dominant, but here it is not be completely dominant, which is why this is exception number 1 to the law of dominance, and this is known as incomplete dominance, children. It is known as incomplete dominance. And if you are asked the genotypic ratio and the phenotypic ratio, over here all are red, so chodo, all are red, Phenotypic ratio is basically one same genotypic ratio. But over here, what is the genotypic ratio and phenotypic ratio? So let's see. I am writing phenotypic ratio first. So I see one is red, the other two are pink and one is white. So one is red, other two are pink and one is white. This is the phenotypic ratio, how we see them. How is the physical appearance, okay? Now what about the genotypic ratio? Genotypic ratio as in the ratio of their genes. So both of them, they are homozygous red. So there is one homozygous red. Then there is heterozygous pink, not heterozygous red, this is heterozygous pink, that is 2. And then there is one homozygous white. So it is 1:2:1. So both phenotypic and genotypic ratio of F2, this is F2, right? First we cross, we got this, then we are self-pollinating them, then we got this, okay? This is the genotypic and phenotypic ratio of F2 plants of snapdragon, and they are showing incomplete dominance. Okay, why again they are showing incomplete dominance? Probably this particular R, when they are single, it's not able to produce enough amount of protein to make the flower red. Instead, there is a white color being already present over there, so it is not completely dominant over the white color. Okay, so that is the thing. I hope you got it. Now let's read NCERT. You'll get more clarity. So, when experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had the phenotype that did not resemble either of the two parents and was in between the two. Okay, over here, if you see, does this represent any of the parents? No, it was in between, right? Now, going ahead, the inheritance of flower color in dog flower. Okay, snapdragon, you also call it dog flower children. Okay, please remember, it is also known as dog flower, not dog. Dog flower, not dragon, okay, and snapdragon or Antirrhinum species as well. Okay, so you have two different names. I'll write it here so that you don't forget. So you have dog flower, or you also call as Antirrhinum species. Okay, 1, 2, 3. Snapdragon, dog flower, Antirrhinum, same thing. Okay, you can make something out of it. ADS, SAD. Okay, you can put sad. Okay, things like that. Das, whatever you want, you can put. Okay, das, sad, whatever. Snapdragon, dog flower, Antirrhinum, all of them are the same. Cool, cool, cool. Now, let's see. The inheritance of the flower color in dog flower is a good example to understand incomplete dominance in a cross between true breeding red flowered and true breeding white flower plants. True breeding, as you know, homozygous condition is right, true breeding. Now, it was found that F1, capital R, small r, are pink in color. Now, when F1 was self-pollinated, the F2 resulted in the following ratio, 1:2:1. Here, the genotypic ratio were exactly as we would expect in any other Mendelian monohybrid cross. Yes, in any other Mendelian cross, for example, when we spoke about this also, over here also, you saw the genotypic ratio was the same, 1:2:1, just like any other. But the phenotypic ratio has changed from 3:1 dominant recessive ratio. So over here, you saw it was 3:1, but now these are not tall. Over here, instead of being red, we have what? Pink children. So it is 1:2:1 in the case of snapdragon. What happened was R was not completely dominant over small r. This made it possible to distinguish RR as pink from capital R as red and small r as white. Now this is given in your NCERT, the picture. They have made a Punnett square out of it. We can also make a Punnett square out of it. So for Punnett square, what do you need, children? We need the gametes on either side. So we are self-crossing F1. So if F1 is there, 1 R, 1 small r, 1 R, 1 small r. Both of them, okay, this is red. When we fuse this, we get this. When you fuse this, you get this. When you fuse this, you get this. So 1 is red, 2 are pink and 1 is white. Okay, this is Punnett square. Given by Reginald C. Punnett, he was a British geneticist. Okay, now this is the first question that we have. In Antirrhinum, a red flower was crossed with a white flower, and in F1 generation, all pink flowers were obtained. When pink flowers were selfed, the F2 generation showed white, red and pink flowers. Choose the incorrect statement from the following. You have to answer me, okay? Law of segregation, incorrect statement you have to. So whenever this type of question comes, please understand what they are looking for. Incorrect. Don't go and search for correct things. Okay, everything is correct now. Law of segregation does not apply in this experiment. This experiment does not follow the principle of dominance. Pink color in F1 is due to incomplete dominance. Ratio of F2 is 1/4, 2/4, 1/4. NEET 2019 question. Okay. So do you think here law of segregation does not follow? Law of segregation means there won't be any blending in cat, as in basically when they segregate from here to here, they segregate, they are going to, for example, you got R and R. Now they are not going to be forever together. These alleles are going to separate and they are going to pass down into each gametes. So each gamete will be having one R and small r. Now they will fuse together and give rise to new things. This is why we still have the parental genotype over here. Okay, which is why we still have the parental genotype over here. Even though little amount of blending is there here and there, but it's not like everywhere. Okay, and this is not exactly blending also. We can say it's incompletely dominant. That is the correct statement. So law of segregation is definitely being followed up here. Okay, but there is another law that is not followed. That is your thing to find out. Now next thing, F2 generation in Mendelian. F2 generation in Mendelian cross showed that both genotypic and phenotypic ratios are same as 1:2:1. It represents a case of what? Codominance, dihybrid cross, monohybrid cross with complete dominance, monohybrid cross with incomplete dominance, 2012. Question. Tell me the answer. Going ahead. Codominance and multiple alleles. Now that is next thing, and for that thing I am very much excited. Listen. So basically, on our blood cells, on the surface of it, we have certain antigens. Okay, agreed? Now, over here, I am saying we have three types of blood groups. Not three types of blood groups, we have four types of blood groups. A, B, AB, and O. These are what, children? Phenotypes. These are four phenotypes of blood. Something that we can see, we know that there are four phenotypes of blood: A, B, AB, and O. Okay, now we have to see their genotype as well. When does A come, when does B come, when does AB come, and when does O come? So please understand one thing. Okay, the gene I will represent as I. Okay, IA stands for giving out A blood group, but the conditions are there, and IB stand for giving out B blood group. Small I, or basically IO, stand for O blood group. Okay, now let's see when can you get this thing. So I am saying when from one parent you are getting IA, from the other parent also you are getting IA. At this point of time, children, you have A blood group. Okay, it is homozygous A condition. IA, A thing. Okay, now from one parent you are getting A, from the other parent you are getting O. At this point also you will get A blood group. Why? So you understood one thing, that basically A is dominant over O. Agreed? A is dominant over O. Okay, now what about B? Let's see. So for having B blood group, again from one parent you should be having IB, from other parent also you can have IB. In this case it will be homozygous B condition. Anyways, you will be having. Next scenario, when one parent you are having B, from other parent you are having O. At this point also there will be sorry there will be B blood group. Now again you might be thinking, how is that possible? Yellow. Again O is recessive. So B is dominant over O. So we got to know A and B both are dominant over O. So now going ahead with O blood group, I want to shift it a bit because we have not put about AB blood group. We have to talk about AB also. When does a person get AB blood group? Let's see. From father, if you get A, okay, from mother, you get B. Okay. At this point of time, you get AB blood group. Ma'am, how can that be possible? Someone has to be dominant over the other, right? Either A or B has to be dominant over the other. But that is a trick, bacha log. What happened over here? They are not dominant over each other. Instead, they are saying, let's be co-dominant. Let's both of us show the sugar that is there on our body, okay, on our cell, whatever sugar is there, okay. For example, A is coding for A sugar, B is coding for B sugar. Now, when a person has A blood group, it is showing both the sugars, okay. So, both of them are saying, let's be co-dominant. But this is not what Mendel thought. This is not what Mendel thought. Mendel said that one has to be dominant over the other. So, this is again exception number 2 to law of dominance, and this exception is known as co-dominance, student. So, this exception is known as co-dominance. That was incomplete dominance over here. This exception is known as co-dominance. But ma'am, now since we have studied about this, can you tell about the other thing also? That is O blood group. Yes, I can. I said if it's I, or it can be represented as IO. Okay. Basically O blood group. So, for being O blood group, you know, if A is there, then O cannot be visible because A is dominant over O. If B is there, then also O cannot be visible. So, for O to be present, so you understood O is a recessive trait. O is what, children? A recessive allele. So, you understood one thing. From both the parents, you have to get O. That's when your blood group will be O. Otherwise, it cannot be possible. Otherwise, it cannot be possible, children. Okay. So, either for AA, you get IA from both the parents and you have A blood group, or IA from one father and from IO from mother, you get A. Similarly, BB from both the parents, B from father, O from mother, again you have still B because B is dominant. But if you're from father and mother, you're getting A and B, then both say we cannot be dominant over each other, we have to be co-dominant. So, that is what you mean by co-dominance, children, and that is the exception of law of dominance. Now comes category number 4, where you want O blood group. Now, O blood group, you know, it's recessive. For that, both the parents should be giving out O alleles, then only things can happen. Now, over here you saw one thing. This is one allele, agreed. This is another allele, agreed. This is another allele. You cannot count this as allele. Basically, this and this comes together to form AB, right? So, this is one allele, this is another allele, this is another allele. For what? For blood group. For blood group, you have 3 alleles, IA, IB and I, or you call it IO. You have 3 alleles, which is why I call it multiple allele condition. Multiple allelic condition or multiple alleles. We say for blood groups we have multiple alleles, children, and we have 4 phenotypes. Okay, A, AB, A, B, AB and O. 4 phenotypes are there. That is different. Alleles are different. Alleles are 3 in number. They come in different combination to give out the different this thing. But phenotypes, if you see, you have 4 of them. Okay, agreed? Can we read it once? Let's see. So, co-dominance happens because no one is ready to give up. A says that I want to show up my protein, I will show up my protein. Similarly, B also says, it's not like Snapdragon. Our body is kind of very much problematic. Now, so till now we were discussing crosses where F1 resembled either of the 2 parents, okay, or was in between. So, either of the 2 parents, as in it was completely dominant, tall or dwarf, tall. Okay, and incomplete dominance where it was showing pink color. No, but in the case of co-dominance, the F1 generation resembles both the parents. Okay, so first it was one parent, first it was neither of the parents, and now it is resembling both the parents. A good example is different type of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by gene I. Name of the gene was what? Gene I. The plasma membrane of the red blood cell has sugar polymers that protrude from its surface, and the kind of sugars is controlled by the gene. So, as I said, from the plasma membrane there are sugars being protruding, and these sugars are controlled by what gene does it have? Does it have IA? Does it have IB? And things like that. Okay. Now, the gene has 3 alleles, IA, IB and small I. The alleles IA and IB produce a slightly different form of sugar, while allele I does not produce any sugar. So, over here, basically, over here, if you see this cell, it will have sugar for IA, okay. If you see this cell, it will have sugar for IB. If you see this cell, it will have sugar for A and B, both. So, this is A and this is B. Talking about this particular gene that is having just I, okay, it does not have any sugar at all. Now, when O blood group is put in some other's body, what will happen? Nothing will happen because it does not have sugar. No other body cells will be able to recognize. If it has sugar and it was different from my body sugar, then obviously everyone will come and attack, which is why we say O blood group is the universal donor. It can donate itself because our body won't be able to recognize. But now it is not done like that. We want specific blood groups, but it was done, okay. O was known as universal donor because it did not have any proteins on its surface, whereas AB was known as universal recipient because it has both A and B. So, anyone can give. Anyways, it has both the proteins. So, not a problem. For our body, it won't be foreign if someone gives A blood group or B blood group. Okay. Sorry, just a second. So, it was known as universal recipient because, yes, if someone is giving A blood group or someone is giving B blood group, either of them, they have the proteins and they also have the protein. So, it's all fine. Okay, so it was known as recipient. It can take anything inside. Now, let's come back to the topic that we were talking about. I don't want you to go there. Yeah. Because humans are diploid organisms, each person possesses any 2 of the 3 I gene alleles. Yes, only 2 we give. 1 chromosome from father, 1 chromosome from mother. Okay, IA and IB completely dominant over I. In other words, when IA and small I are present, only IA expresses because a small I does not produce any sugar. As I said, O blood group comes like that, okay. Now, when IB and small I are present, IB expresses, but when IA and IB are present together, they both express their own types of sugars. This is because of co-dominance. Hence, red blood cells have both A and B type of sugars. Since there are 3 different alleles, IB, IA, IB and small I, there are 6 different combinations of these 3 alleles that are possible, and therefore a total of 6 different genotypes of the human ABO blood group. So, let's see, do we have 6 different genotypes? 1, 2, 3, 4, 5, 6. We have 6 different genotypes that is possible over here. 6 different combinations that can happen in our whole body. In our body, basically in human body, whatever blood group we are having. So, in total, if you talk about blood groups, the total genetic combinations are what I am writing. So, basically it has 3 alleles, which is why it is known as multiple alleles. How many phenotypes are there in total? 4 phenotypes. Either you can be A, either you can be B, either you can be AB, or either you can be O. Now, if you talk about how much total amount of genetic combination or genotype combination are possible, total genotype possible, okay, genotype combination possible I am writing. What will be the total genotype possible, genotype combination possible, children? 6. You can have 6 genotype. IA, IAO, IB, IBO, IAIB, IO. So, basically you have 6 possible genotypes. Please write it somewhere. 3 alleles, multiple alleles, 4 phenotypes possible, 4 genotype combination possible, that is 6, sorry not 4, 6 possible genotypes possible, and that is written over here. Got it? Now, okay, why it does not work? So, over here the same thing they have written, same, nothing is different. Whatever I taught you is the same thing from there only, okay. So, either mother can give you, the father can give you B, either way, if you have both A and B, it gives you AB, okay. Now, can I give you a question? Can you try this question, children? If I say, yes, if I say father is having O blood group, mother is having AB blood group, then what is the blood group of the children? That is possible blood groups of the children which would be formed. Can you tell me the answer? Can you form? For father to be O, what should be the genotype? Should be IO, or basically just small I, okay? I'm not writing like that, I'm writing IO. For a mother to be AB, it should be IA and IB. Now, if you fuse this with this, you get IAO. If you fuse this with this, you get IBIO. If you fuse this with this, you get IA, again IO. If you fuse this with this, you get IB, IO. None of them are O positive. Here A is there to dominate, so it will be A blood group. Here B is there to dominate, it will be B blood group. Here A is dominating, so again A blood group. Here B is dominating, again B blood group. Okay, now a condition. Children. If, can I remove it? If you want, you can take a screenshot of this, okay? Second condition is where, okay, I'll just remove this and put the next one. Father is again O, mother is A blood group. Then what? What are the different types of progenies, genotypes that you can observe in the progenies there? Can we try this? So father is O, so O for it has to be recessive always. So it has to be homozygous like this. Same type of allele should be coming. What about the mother? Mother can be like this. Agreed? Mother can be like this, or father anyways it is this. Mother can be like this as well, because A is dominant over O. So anyways, if she has this genotype, still she would be A positive, sorry, A blood group, she would be having A blood group as well, right? Agree? Now let's see the combination. So over here, this and this fuses, I get AO, I get A and O. If this and this fuses, again I get A and I get O. If this and this fuses, A, O, this and this fuses, again A, O. All of them, children, it will be A blood group. All of them. No chance of something else. But over here, this and this fuses, IA, IO. A is dominant, so you get A. If this and this fuses, you get IO, IO. So it will be O blood group. If this and this fuses, again IA, IO, that means A blood group. If this and this fuses, you get IO, IO, that is O blood group. So you see, you have to be very, so if they are asking you that a person having A blood group can have a genotype like this, can have a genotype like this, it's true. Both the genotypes are possible if they are A blood group. Similar thing with the B blood group as well. It can be IB, or it can be IB, IO. Got it? Will you be able to do these questions? Yeah? Now, going ahead, children. So this is the combination. Now over here I have a question. I think this is question number 3. No, question number 4. A gene showing codominance has alleles that are recessive to each other, both alleles independently expressed in heterozygote, one allele dominant on the other, alleles tightly linked on the same chromosome. This is 4th number question. Do let me know the answers in the comment section. Okay, I want to know your answers before I start with something new. Now. Next thing, 5th. Ayo, in that case, this is 3rd question. Wait. Just a second, okay? I'm a little confused. Did we repeat this question? 1, 2, 3, 4, 5. Okay. This is 3rd question. Please say 3rd question and then answer. This is 3rd question, okay? Now going ahead with the fourth question. Genotype of a husband and wife are IA, IB and IA, IO. Among the blood types of the children, how many different genotypes and phenotypes are possible? Genotypes and phenotypes are possible. You have to solve it. Solve it by yourself. Take a screenshot, pause the video and do it. Okay, I'm not going anywhere. Okay, you can continue the video later on once you're done with this. But I'll be proceeding with my next question. Okay, solve this, please solve this and do let me know now only. Identify the wrong statement with reference to the gene I that controls AB blood group. Okay, the gene I has 3 alleles. A person will have only 2 of the 3 alleles. When IA and IB are present together, they express same type of sugar. Allele I does not produce any sugar. Okay, again, can you tell me the answer for the same? Is there? Can you solve directly from your NCERT? It's simple. I'll solve at the end. Now there is a term, as I said, that is pleiotropy. What do you mean by pleiotropy? Basically it means a single gene is going to decide multiple traits. A single gene, as of now we saw that capital T was giving tall height and small t was giving dwarf height. But I am now saying that capital T will be also giving rise to tall height and something else. So it's not the example. I'll just shift the example to something else that is given in your book, which we have to study. Chalo. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. So I am saying that starch synthesis is controlled by one gene which has 2 alleles, okay, and that gene is capital P, and it has 2 alleles, so capital B and then it will be small b. Okay, starch is synthesized effectively by BB homozygotes and therefore large starch grains are produced. In contrast, okay, listen to me very carefully, okay, this is responsible for round seeds. Okay, this is responsible for round seeds, whereas this is responsible for wrinkled seeds. Now children, when you cross capital B, small p, what do you get? When this and this cross, you get capital B, small b. When this and this cross, you again get capital B, small b, same thing. Capital B, small b, capital B, small b. How many of them? 4 of them. Now, over here, all the 4 will be round. All the 4, they are round. Okay? Agreed? But there's a small challenge over here. That is, when condition is like this, it is obviously round, but the amount of starch which is there, it is way too much. So it is round, and starch content is basically way too much. When condition is like this, still it is round, but capital B is responsible for one more thing, that is for the formation of starch, and this would produce less starch, whereas this produces more starch. So when 2B are there, they are producing enough amount of starch. When 1B is there, it produces little amount of starch. Okay, did you get it? Okay, so here the size will be intermediate, here the size will be very big. Okay, now talking about small b, this will be wrinkled and the starch is very little. This will be wrinkled as well as the starch is very little. So over here you are seeing one very important thing, that the allele B, okay, the alleles or basically the gene B is controlling 2 different traits. it is also looking for the shape of the seed. It is looking for the shape of the seed as well as the amount of starch. So when it's capital T, capital B, obviously it will be round, but the starch content will also be very much because it is also looking for starch. When it is capital B, small b, at this point of time, the starch content is comparatively lesser. The starch is comparatively lesser because this is also responsible for starch formation, right? And here less amount of starch is being formed, which is why this will be of intermediate size even though it's round. Talking about small b, it will be wrinkled and the starch is comparatively very, very less. Did you understand this? Okay. And also one more thing. Can I say that this is incompletely dominant over small b? When these are present, either it has to be very round, it has to be having enough amount of starch, but that is not being possible over here. Okay, round you live, but at least amount of starch. The amount of starch being produced is very intermediate level, right? So if B was there, probably it should have shown both, effect. It should have been completely dominant and should have been showing round and also should have been producing enough amount of starch, but that is not happening. It is producing very little amount of starch as in comparison to that. So basically this is showing incomplete dominant. If it was completely dominant, then probably it would have had enough amount of starch as well. So this is an example of pleiotropy where a single gene is controlling multiple traits. While it is doing it, we came across one more thing, that since enough amount of starch is not being produced in heterozygote B, this means it is incompletely dominant as well. Okay, so two things you got from this only. I hope this is clear to you. Let's read it once. So over here, starch is synthesized effectively by capital B homozygotes. Therefore, large starch grains are produced over here. Okay? In contrast, small b homozygotes have lesser efficiency in starch synthesis and produces smaller starch grains, as you can see over here. After maturation of the seed, capital B seeds are round and small b seeds are wrinkled. Heterozygotes produce round seeds and so B seems to be the dominant allele. So this is round, so this is dominant. For this particular trait, capital B is dominant. But what about starch? For starch synthesis, it is showing incomplete dominance. Because the amount of starch being produced is intermediate. But starch grain produced are of intermediate size in the BB cell, that is capital B small b cell. So if starch grain size is considered as phenotype, then from this angle the alleles show incomplete dominance. So in this angle it is showing incomplete dominance because you are getting intermediate sized one. But over here if you talk about the shape, that is showing complete dominance. Agreed? So if you are asked the example where the things are showing complete dominance as well as incomplete dominance both. Okay, but the phenotype is different. Phenotype is something different. Okay, but with the same genotype is showing incomplete and complete dominance. So basically I can give the example of this. Okay, that is of the seed shape as well as the starch content. In the case of starch content, it is showing incomplete dominance in heterozygote. In the case of shape of the seed, it is showing complete dominance as well. This is known as pleiotropy, children. Please remember it has came in 2023. Okay, single gene, multiple traits. Now let's solve the questions which were there. Okay, I have already explained, so we can do the questions over here. In antirrhinum, that is snapdragon, or what do you call it, children? Dog flower. A red flower was crossed with a white flower, and in F1 generation all pink flowers were obtained. When pink flowers were selfed, the F2 generation showed white, red and pink flowers. Choose the incorrect statement. Law of segregation does not apply in this experiment. Law of segregation definitely applies, which is why you have red and white colors still being formed. Okay, in F2 generation. This experiment does not follow the principle of dominance. Yes, this does not follow the principle of dominance. That is why it is showing co-dominance. It is not completely dominant. According to law of dominance, one in heterozygote, in a dissimilar pair of factors, one should be dominant. But over here, it's not like that. Pink color in F1 is due to incomplete dominance. Yes, children, it is due to incomplete dominance. Ratio of F2 is this. This is also correct. So this is the problematic thing. Now, genes showing codominance. Okay, the genes that shows codominance, alleles has alleles that are recessive to each other. No, codominance, as in both of them, they are showing together they're proteins, so they cannot be recessive. Both alleles independently expressed in heterozygote, yes. So heterozygote as in when the alleles are not same. So IA, IB, this is in heterozygote. Here the alleles are not same, but still they are independently expressing themselves. So this is correct. One allele dominant on the other, no. If they were dominant, obviously it will show complete dominance. Alleles tightly linked on the same chromosome, no children, this is the answer. Got it? Now, F2 generation in a Mendelian cross showed that both genotype and phenotypic ratio are the same as 1:2:1. In where? We saw this in monohybrid cross with incomplete dominance. If you are not remembering it, I'll show you. I think here we drew. Okay, here you see 1:2:1 genotypic and phenotypic ratio we saw, right? Red, pink and white, and also capital R, small r and small r. In F2 generation it was shown in monohybrid cross. We are talking about just one trait over here, that is the flower color, and it is showing incomplete dominance. So let's go back over here. This is the correct answer. Okay, the genotypes of husband and wife are IA, IB and IA. Okay, for this we have to make it. So let's, are you ready to make it? So what is it? IA, IB. Okay. IA, IB and then IA and IO, or basically I, small I they have mentioned. Now they are asking the combination. So let's see. It can be IA, IA, it can be IA, IO, it can be IA, IB, it can be IB, IO. Genotypes, if you see, all of them, they are different. It's AA, it's AO, it's AB, it's BO. All of them are different. So we have how many genotypes, children? We have 4 genotypes. Phenotypes, if you see, let's see. Phenotypes as in how, what is the actual product that they are going to give. This is A blood group. This is again AO, means A is dominant over O. So this is again A blood group. This is AB blood group and this will be B blood group. So phenotype, I see 1 phenotype, 2 phenotype and 3 phenotype. So we have 3 phenotypes. Calculate this to be 1 only because it's the same thing, no? A blood group. So 1, 2, 3. 3 phenotypes, 4 genotypes. Let's see what matches our answer. 4 genotypes, 3 phenotypes. That is the answer, my darling children. So over here, please put. Okay. Now identify the wrong statement with reference to the gene I that controls ABO blood group. So gene I that controls ABO blood group has 3 alleles, yes, A, B and O. The person will have only 2 of the 3 alleles, yes, from one parent, from other parent. So 2 copies. Okay, fine, done. When IA and IB are present together, they express same type of sugar? No, they show different type of sugar, and both the sugars are there, which is why we say they are co-dominant over each other. Allele I does not produce any sugar? Yes, small I, allele small I does not produce any sugar. Basically IO is not producing any sugar. It is this statement that is what? Incorrect. They are not expressing same type of sugar, they are expressing different type of sugar. Okay. Now going ahead, children, what do we have? One question that I have for you is this thing. Try this question out. ABO blood grouping is controlled by gene I, which has 3 alleles and show codominance. There are 6 genotypes. How many phenotypes in all are possible over here? Let's try it out. We have studied this, and I hope that this particular session that we took was really helpful. Even I had fun teaching you guys. I hope you also had fun learning with me. Now if you have any doubts regarding this, I am here. You can comment your doubts down. I'll be reading down your comments and trying to give you the answers as well. And also if you have got all the answers correct by doing it yourself, do let me know. I'll be really happy seeing that as well. I hope that you just take good care of yourself and be happy. Do not forget to subscribe to the channel. Bye bye children. Take care.